Tetrodotoxin, as shown on the slide, is a neurotoxin found in multiple animal species, such as blue-ringed octopuses, pufferfish, frogs, and my favorite, newts. TTX, as it's known for short, works by blocking sodium channels on nerve cells, therefore causing paralysis and eventually asphyxiation. A rather slow and arduous way to go out, if you ask me, but definitely an interesting one. My research concerns TTX in newts, specifically the rough-skinned newt, properly known as Pterica granulosa, like the one seen on the slide. Newts are salamanders, and this specific newt gets its name from its rough dorsal surface, which contains little TTX glands. We still don't know where or how TTX is produced in newts, but we do know that it is found on its dorsal surface, as well as in internal organs such as the ovaries. It has been suggested that the levels of TTX in the ovaries are under active maternal control. Essentially, this means that female newts are able to control the levels of TTX in the ovaries and/or control the transport of TTX within their bodies. If TTX is being transported within female newts, we should be able to detect changes in TTX levels in different parts of the newt in response to ovulation. I hypothesize that during ovulation, TTX is transported to the ovaries of the female newts from their dorsal surface. To test this, I'll be injecting female newts with LHRH, a hormone that's used to trigger ovulation. Tetrodotoxin levels on the dorsal surface of newts will be measured by taking skin punches before and after injecting the newt with LHRH. TTX will then be measured and the levels of TTX will be compared before and after injection. A control group will also be set up where newts will be injected with saline instead of LHRH to control for any stress that could be associated with inoculation, handling, etc. By conducting this study, I hope to find that dorsal TTX levels in newts decrease in response to ovulation, which may suggest that TTX is being transported from the dorsal surface to other parts of the newt, such as the ovaries. This work will allow us to better understand the evolution of tetrodotoxin, as well as the physiology and ecology of the rough-skinned newt. Thank you.
Hunters of the night, they awake as the sun goes down and fly through the darkness in search of prey. No, we're not talking about vampires. We're talking about bats. Bats comprise 1/5 of all mammal species, yet they are often misunderstood by the public. Bats are ecosystem indicators, meaning that their presence or absence implies the conditions of their habitat. Importantly, we can use bats as a proxy for determining the recovery of habitats following natural disasters like fire. While wildfires are a natural part of California's landscape, the fire season in California and across the West is starting earlier and ending later each year, with climate change considered to be the key driver of this trend. The purpose of my study is to understand how bats respond to fire-affected habitats. Specifically, the aim of this project is to determine, 1, how changes to density of vegetation from fire affects the activity patterns of forest-dwelling bats, and 2, how the number of bat species in a habitat changes over time due to the degree of fire severity. To evaluate the effects of burn severity on bats, we conducted acoustic surveys this summer within the SQF complex that burned in Sequoia National Forest and adjacent areas in August 2020. Acoustic monitoring is a non-invasive method that allows us to record echolocation calls, which can be used to identify different bat species by their unique echolocation signatures. 17 of the 23 bat species in California use Sierra Nevada habitats. 11 of the 17 expected in Sierra Nevada parks have special status designated by a federal, state, or other organization. Sonobat software allows us to record, process, and analyze acoustic data from bat echolocation calls. And this can be seen on the top of my slide where I have a call from a Yuma myotis bat. Our Sonobat results show that the most commonly detected species included the Mexican free-tailed, California myotis, pallid, and Yuma myotis bats. With those species in mind, we know that fires alter bat foraging habitat structure by reducing clutter in the form of tree and understory vegetation density. This change causes detriment to clutter-adapted species, such as the California myotis. My preliminary results show most California myotis calls occurring in unburnt habitat and habitat with the lowest fire severity. Fire influences animals through direct mortality and indirectly through habitat alteration. Therefore, this information is crucial in understanding how bats are adapting to fires, encouraging appropriate habitat management and conservation efforts of an order already facing numerous threats. Thank you for your time.
We've probably all heard about the plastic straw stuck in the sea turtle's nose, or the massive patches of garbage floating in our oceans. The global production of plastics alone is approaching 400 million tons each year, with only about 9% being recycled. While the durable nature of plastics is appealing for practical use, this is also the reason why plastics persist in the environment for long periods of time. Because of this, man-made materials are unfortunately a permanent component of our natural world. While natural leaf litter is an important source of energy and nutrients for terrestrial and aquatic systems, plastic litter, on the other hand, can be harmful for the ecosystem and the animals living in it. Understanding how long plastic waste persists once it enters the environment is crucial to determine the fate of these materials, which is important to estimate global plastic budgets and target management efforts towards sensitive ecosystems and organisms. The goal of my project is to determine if the rate of plastic degradation in freshwater and terrestrial habitats is different compared to natural tree leaves. To accomplish this, we used a traditional leaf pack experiment using 3 different treatments, which you can see in the top left corner of my slide. Mesh bags were filled with natural tree leaves, a common plastic litter, which we used red plastic bags, and a mix of the two. 50 of each treatment were anchored in a terrestrial grassland, which is seen on the bottom left corner of my slide, and in a freshwater pond, which you can see on the top right. Subsets of the leaf packs were collected every 2 to 6 weeks, along with any macroinvertebrates that may have made a home out of the leaf packs over time. The contents were weighed before and after they were collected to measure degradation rates through time. Data for this project is still being collected, with some leaf packs still degrading in the pond and the grassland habitat, so no statistical results are available at this moment. However, I do predict that the plastic will end up degrading faster in the grassland due to high temperatures and direct, intense UV exposure that's known to break down plastic materials. I, for one, sure know that I feel like breaking down when I'm outside in that summer heat. I also predict that the natural leaves will break down faster in the pond due to constant water submersion, since moisture is an important variable for the degradation of natural leaf litter. Future results concluded from this study will contribute towards knowledge on how long plastics persist in the environment compared to naturally occurring materials. And this will provide a deeper understanding into the life cycle of plastics that will facilitate management strategies for reducing plastic pollution in our environment. Thank you for your time.
Hello, everyone. Valley fever is a fungal disease caused by soil-borne pathogens Coccidioides mitis and Coccidioides posidosii. And while there's plenty of information readily available explaining this fungi and its pathogenicity, how it potentially causes upper respiratory distress and systemic organ failure, and even general areas in which it resides, there's very little information explaining the relationship between valley fever infections and marine mammals, and even less explaining how valley fever is being found in areas once known to be inhabitable for its pathogen. So if we look at figure 1, the graph indicating valley fever incidence in coastal counties, we see the trend line periodically increasing up until 2017, when there is a massive increase in reported cases. Now, this is quite interesting when we take into consideration that valley fever tends to prefer warm, dry, even hot soils and temperatures compared to what we find in these coastal California counties. So now, what's contributing to valley fever being found in areas once not known to find, and what's contributing to the introduction to marine populations? So moving into figure 2, we see that snapshot of southwestern United States. These red dots are indicative of known confirmed valley fever fungal hotspots, and the black arrows indicating the directionality of the Santa Ana winds, which have the tendency to uplift small soil and dust particles and carry them away from their origin. So now we see that these winds are depositing these soil samples along coastal counties, but also in coastal waters, as indicated by that white region that covers the Channel Islands. These waters off the coast are where marine mammals such as sea lions and whales, dolphins tend to roam, migrate, and breed with one another. So their close proximity to fungal-rich soils, as well as around and in fungal-rich airs, can be the first introduction of this fever into the marine mammal species. But is there anything else contributing to why it's traveling further? So that's where my research took me to Catalina Island this summer, as we can see a snapshot in figure 3 in the top right, which is Two Harbors, a small village on Catalina Island, where I collected soil samples as indicated by the blue dots. These soil samples were collected on a trail that transected differentiating soil types to increase the diversification of soil types, as well as to increase the likelihood of finding valley fever fungal spores, if any present. So now once these samples were collected and taken back to the lab, compared all DNA found in the soil to known DNA of valley fever. So if we see in figure 4 in the bottom right, the green lines pointing to those very darkened bands are confirmed indications that valley fever from mainland is present on the Channel Islands, specifically Catalina Island. So now what's to say, is it the marine mammals and their breeding migratory patterns? Is it the Santa Ana winds and their directionality? Or is it something in between, potentially both? This further constitutes research to look into potential causes for how this is contributing to the declination of marine species. Thank you.
I've been a nurse for 5 years. All my time working as a RN has been in the acute care setting. A majority of the patients that I encountered were non-compliant and developed complications from chronic conditions such as hypertension, diabetes, and heart disease. Although there are many different diseases that deserve some spotlight, I decided to stick with diabetes for 2 reasons. First, out of all the days that I've worked, I've had at least one diabetic patient. My second reason is that diabetes is becoming more common in children and adolescents today. I'm sure everyone here knows of at least one person who has diabetes. I mean, diabetes is one of the top 10 leading causes of death in America today. Research by Lynch and others showed that educating diabetic patients results in these patients walking away with more knowledge about how to manage their diabetes when compared to the initiation of the study. So I asked myself, are nurses educating these diabetic patients compared to these patients that are admitted to the hospital for complications related to diabetes in terms of compliance, diet, exercise, and prevention of complications? First, on medication, when someone has an infection, they are given antibiotics and the infection typically resolves. Sometimes only medications are required to manage a disease or illness. A diagnosis of diabetes is not just taking medication, but requires a lifestyle change. Second, diabetic patients need to eat healthy by avoiding bad carbs and eating more vegetables. Third, daily exercise needs to be initiated, even if it is mild exercise. Lastly, taking preventative measures such as foot care and blood sugar checks to prevent any complications. Consequently, in this study, we tried to determine if nurses are providing comprehensive diabetic education, recognize any barriers that hinder education, and being mindful of the type of education is being provided to patients. So a quantitative 20-question multiple choice survey was completed by 47 RNs in the Bakersfield area. Approximately only 1/3 of the surveyors reported providing education during encounters with diabetic patients. Roughly 1/4 of the nurses provide education about exercise, and 1/3 surveyors reported education about diet, medication compliance, and preventative measures. The most common barrier recognized was time constraint. There needs to be some kind of adjustment so that nurses and providers have time to properly educate patients. Thus, this brief study demonstrates that nurse-patient ratios, record-keeping requirements, and workload hinders patient diabetic education by nurses. Thank you.
Did you know that some animals share characteristics with one of the things that parents give out as party favors at kids' birthday parties? And no, I'm not talking about candy, bubbles, or those little tubes that you blow on that shoot that paper out that kids love and adults find annoying. No, I'm actually talking about glow sticks. You heard that right, glow sticks, referred to as biofluorescence when talking about animals. Biofluorescence is most seen in aquatic organisms, but has also been observed in terrestrial organisms like spring hares, scorpions, and even flying squirrels. So, what is biofluorescence? Biofluorescence occurs when a living organism and then reflects that light at a low energy, which results in a glow that can occur in different colors. The presence of biofluorescence in amphibians like newts has not been studied at great length. Biofluorescence has been shown to be present in other amphibians, but the reason behind such mechanisms is not widely known. For my research, I tested for biofluorescence in the rough-skinned newt Turica granulosa. I did this by shining a blue light similar to a UV light on the newt. I then photographed the newt with the filter. And something amazing happened. The newts glowed. My preliminary research has shown a pattern of biofluorescence along the limbs of the newt and concentrated on the belly of the newt. The next step in my research would be to figure out why and how the newts have biofluorescence. Some studies have indicated that biofluorescence in amphibians was likely present in their early evolution. Biofluorescence may be used for communication between individuals of one species or between individuals of different species. This mechanism may be helpful for things like camouflage, finding mates, or it might have another purpose that we don't know about altogether. Newts might also fluoresce due to pigments in their skin or because of the presence of fluorescent proteins, though such proteins have yet to be identified in amphibians. So the big question here is, why does this research matter? Well, amphibians have had big declines in population sizes because of things like habitat loss and climate change. Unfortunately, we still don't know a lot about them and how they maintain healthy populations. Understanding biofluorescence may help us better understand how newts reproduce and find mates or protect themselves from predation, which may in turn help us better understand how to protect their habitats. Also, it's awesome. Thank you.
I'm a nurse at Kern Medical in the emergency department, and I've seen plenty of COVID-19 over the last 2 years. We're all familiar with the symptoms of COVID-19, that being fever, chills, cough, shortness of breath, nasal congestion, nausea, diarrhea, so on and so forth. What we aren't quite as familiar with are the symptoms of a pandemic, a global pandemic at that. You're not alone if you have recently felt a sense of overwhelming stress and anxiety related to COVID-19. In fact, SARS-CoV-2 is responsible for the pandemic that has negatively affected the mental health of humans across the world. Here are some fun, not so fun facts about the COVID pandemic and its impact on mental health. 53% of people report that the pandemic moderately or severely impacted their mental health in a negative way due to consequences such as death of a loved one, lack of basic needs, and lack of normal social interaction. Citizens aged 25 to 49 were found to have an increase in their alcohol consumption in order to cope with the difficulty of the pandemic. Within this age group, parents of young children have been uniquely affected due to working from home, educating, as well as entertaining their children and caring for their families. During the COVID-19 pandemic, families have been faced with a major lifestyle change, financial hardships, issues with childcare, and having to do all of these while monitoring your 5-year-old child pouring glue on their laptop during a Zoom class. And yes, I am speaking from experience on that last one. As a result of the restrictions and precautions associated with mitigating the spread of this mysterious virus, patients have lacked the resources necessary to adjust to life, with children who have been kept from school, daycare, recreational activities, which has placed significant stress on their jobs, relationships, and mental health of both parent and child. The purpose of this research is to investigate the levels of self-perceived stress among adults during the COVID-19 pandemic. We hope to uncover specific demographics in need of mental health support, rehabilitation to provide targeted, specialized intervention, and tailored mental health care. Additionally, this acts as a retrospective research study to prepare mental health care workers, mental health care providers for the inevitability of future pandemics. For our study, 169 participants aged 18 to 65 completed a 20-question online survey, and their results were quantified using a novel screening tool developed by the authors, the Self-Perceived Anxiety and Stress Tool, or nicknamed SPAS. Among the subjects who responded, females scored significantly higher on the SPAS when compared to males, indicating that females reported significantly higher levels of self-perceived anxiety and stress. Additionally, there was a significant difference among age groups, with the highest stress and anxiety reported in the age groups 25 to 34 and 35 to 44, consistent with previous research. Employed respondents were significantly less stressed when compared to unemployed individuals. Additionally, there was no significant difference between parents of children that attended in-person versus online learning or distance learning. The results of this survey indicate that females, those aged 25 to 44, and unemployed individuals were most in need of mental health services. Further studies should include larger randomized control trials determining the implications of a global pandemic. What's important to know and take from all of this is that anxiety is not wrong. Anxiety is simply an emotional response to a perceived future threat. These times are unprecedented, and navigating these feelings of anxiety are very new and seemingly uncontrollable. So just remember, you are not alone, you are not wrong, and it's okay to not be okay. Thank you for your time.
Rough-skinned newts are amphibians in the salamander family. Their name comes from the rough texture on their skin. These newts have evolved a powerful neurotoxin called tetrodotoxin, or TTX for short. TTX works by shutting down sodium channels in cells and leads to paralysis. Newts may have evolved this defense in response to predation and parasitism. More recently, however, TTX in newts forms a geographic mosaic pattern that mirrors the resistance of their main predator, the garter snake. TTX in rough-skinned newts varies widely across and within populations. It can range from undetectable to 27 milligrams, enough to kill 50 full-grown adults. Newts have a distinct coloration on their ventral or their belly side. We call this a warning coloration, and predators learn and recognize that these colors mean stay away and poisonous. Newts possess this coloration honestly, meaning they should in fact not be eaten. Because this coloration is on their ventral side, it is not immediately apparent to a potential predator. They need a way to display it. Meet the Unken reflex. The Unken reflex is a defensive posture found in amphibians used to warn predators of toxins. Unken in present forms is various across many amphibian families, including the fire-bellied toad, Bombina bombina, for which it is named. The Unken reflex varies in degree of the reflex both across species and within newts, as you can see with the 3 newts shown here. What we do know about Unken in newts is somewhat sparse. What we do know is that it is a cube that predators respond to. It has been shown to deter predators even in the absence of coloration. What we don't know ranges from questions about whether less toxic newts will enter Unken, if there are optimal conditions for it, if there are predators they'll go into Unken for, but not for others. I could go on. What I'm focusing on is if there are temperatures that favor greater degrees of Unken or a greater likelihood of entering the pose. At what point of the threat they'll enter Unken, whether or not it's mating season, especially if they're gravid, which means carrying eggs, and what role hormones may play in degree of Unken. Adding to our knowledge of this defining but poorly understood behavior collectively enhances our knowledge of the seedy and abundant animal. Thank you.
The spread of the coronavirus was declared a global pandemic on March 12, 2020. Education institutions around the world made an abrupt shift to pivot their in-person courses to an online medium. Prior to the pandemic, Bo and colleagues estimated in the United States that 20% of teachers will resign before their 3rd year of teaching and 50% by their 5th year. In April 2021, more than 4 million employees quit their jobs in the US, and another 4 million quit their jobs in June. Granderson calls this time the Great Resignation of 2021. The impact the pandemic brought on teachers, students, and colleges from switching many of their classes to an online medium is not yet fully understood. Ferry and colleagues determined that technology, pedagogy, and social challenges are the three main obstacles faculty would face as new online instructors. First, technology challenges included internet connectivity issues, training, and access. Second, new pedagogies are needed for online teaching to keep students engaged and to implement innovative online teaching activities. And third, social challenges include the loss of human interaction between teachers and their students. These challenges may lead to some faculty to experience burnout, prompting their decision to retire early or resign from teaching. However, Green and colleagues argue that faculty retention is important to maintain academic continuity, avoid costly turnover, and prevent damaging its reputation. The California public higher education systems may have suffered a significant increase in faculty employment attrition rates during the pandemic. Faculty groups by tenure status, race, and sex need to be investigated to determine if certain groups were impacted more than others. It is crucial to understand if the Great Resignation has impacted higher education. Preparations for future emergency teaching begins with defining the situation, determining if there's a problem, and providing plausible solutions, which is the aim of my research. Thank you.
Hi, everyone. I'm Jenny, and I am an emergency nurse here in town. And we wanted to do our project on burnout and compassion fatigue in healthcare workers. So the purpose of our research was to see if there were higher rates of burnout and compassion fatigue in healthcare workers that work in COVID units versus non-COVID units. And then we also wanted to see if there were higher rates of burnout and compassion fatigue before the pandemic versus after the pandemic. So you might be asking yourself, what is burnout? Burnout is environmentally driven. It's due to time constraints, resource constraints, increased patient-nurse workload. Compassion fatigue is when healthcare workers are not able to save their patient from harm. So they end up having guilt and distress, wondering what could have they done better to take care of them. So research has shown that a combination of limited resources, inadequate staffing, lack of administrative support, increased patient acuity are all factors that increase burnout and compassion fatigue. So it's really important to research burnout and compassion fatigue because it impacts healthcare workers and nurses severely. Research has shown that emotional burden can affect patient care. So if healthcare workers and nurses cannot take care of themselves, how can they take care of patients? So we came up with a 21 question survey on Qualtrics that was available for 4 weeks. Our results showed that 130 healthcare workers responded. About 88 actually worked in a COVID unit. The main concern was that they were afraid of bringing COVID home to their family and friends. And you can see in the bar graph here, other concerns were getting COVID themselves and running out of PPE. Our results also showed that there are higher rates of burnout and compassion fatigue in healthcare workers that work in COVID units. And we also found out that burnout increased from 50% before the pandemic, and it is now at 98% during the pandemic. And compassion fatigue rates increased from 26% before COVID and is now up to 33% during COVID. So although nurses are burnt out and suffering from compassion fatigue, We're committed to the profession and we'll always be here to take care of our patients. Thank you.
The person who wins the competition today will get this beautiful clock. In the future, we'll plan to engrave your name. So apologies that we're still learning. So it's a beautiful clock. In addition to that, you will also get a nice certificate of participation signed by me. You know, maybe I could get the president. I'll see if someone of importance, you know, that to sign it. But it's just a gratitude for the investment that you put into these projects. And I have the ratings, and it's going to be, we're going to announce again, the person who wins is going to go to the Western Association of Graduate Schools in Colorado. I believe if we're able to go in person, the university will pay for you to go and participate there. So that will be the first person. And then in May, we're going to have a conference with all the Cal States, and we're allowed to send 2 people, okay? So first, we're going to announce the second place person. Let me say that again, the scores are within a small margin. So all of you guys did terrific work. So let me.
All righty. For second place, we have Miss Bella Ozuna. Congratulations, Bella.
Yay, thank you. Let's find out who is the person who won the competition today. Raji, are you able to give us that information, please?
Yes, yes. This was, everyone did a wonderful job. You all should be very proud of yourselves as a former bio student. I remember my days at CSUB doing research on fat rats, and that's a whole different story. But congratulations to the first place winner, Autumn Corrow. Hope you're saying your name correctly. Congratulations, Autumn.